The following presentation is on nanoparticles for cancer drug delivery. I am Nathan Costa, your presenter. Let's start with some background. In the year 2012, cancer accounted for roughly 8.2 million deaths worldwide. Specifically in the United States, in the year 2013, the cancer mortality rate was hovering around 20%. Current treatments for cancer rely on radiation and chemotherapeutic agents. These work by destroying rapidly dividing cells in the body, which is the main property of neoplastic and cancerous cells. For these reasons, this is why chemotherapeutic treatments can damage normal, healthy cells that divide quickly in the body, such as cells for bone marrow, macrophages, the digestive tract, and cells in hair follicles. The main drawback of conventional chemotherapy is that it cannot give selective action only to the cancerous cells. They kill healthy cells, and they cause increased systemic toxicity to the patient. So what we need is some form of targeted drug delivery and controlled release of these drugs. How can we do that? Well, we can use nanoparticles. Nanotherapeutics are being implemented to overcome many of the limitations of conventional drug delivery systems. As you can see from some of these figures, the trends in patent filing, and in the table below, we have listed a lot of the approved clinical drugs already that are based on nanoparticle technologies. So, in summary, nanoparticles for therapeutics and even diagnostics have been approved for clinical applications, and there are many that are still currently under clinical trials. Moving on, some of the limitations of conventional therapeutics are listed here. The biggest one being poor water solubility. In relation to that, because they have poor solubility properties, conventional therapeutics tend to aggregate in the body and don't have great biodistribution. As already mentioned, conventional therapeutics also lack selectivity toward cancer cells. They also target normal cells, and again can cause increased systemic toxicity. They don't have any non-specific, no local biodistribution, no control over local biodistribution. They have a low therapeutic index for the most part, and this requires high drug doses to be administered into the patient, the cancer patient. Also, multi-drug resistance is a problem. So, continuously using these conventional therapeutics, the cancer cells in the body actually develop resistance to these drugs, and so new cocktails have to be made up to continue treating the patient effectively. Another limitation of these therapeutics is that they are susceptible to physiological degradation in the body. They have low circulation half-lives, and because of these factors, they have to be repeatedly administered in the body, which again can increase systemic toxicity, as we talked about. So some of the attractive properties of nanoparticles, what makes them so attractive? Well, for one, they're small. They have larger surface area to volume ratio. This allows them to increase their solubility in the solution, in the blood plasma, for instance. Their outer surfaces can also be functionalized or modified to target specific cancer cells. They have tunable size and shape. They can be from anywhere between 1 nanometer we're talking about to several hundred nanometers. They can be easily designed and prepared. We see in this photo here, this is a polymer micelle that's used for drug delivery applications. You simply start with some amphiphilic molecule consisting of a block of polymer, two different blocks, one hydrophobic block and a hydrophilic block. You place them in a solution, and they self-assemble around the drug that you have in their solution, and that's how you can create these micellar structures that encapsulate therapeutics for cancer treatment. Another attractive properties of some MPs are that they are biocompatible and biodegradable. They have increased stability in the body, and that can be enhanced through some cross-linking, particularly for the polymeric micelles used for drug delivery. And of course, one of the biggest advantages of these nanoparticles is that they can be designed to be sensitive to certain external stimuli, such as light, such as like pH gradients, temperature gradients, through the application of electric fields, magnetic fields. So a lot of possibilities are out there. And there are also a variety of architecturals and material formats that can be used to create different drug delivery systems. So some of the advantages of NPs, nanoparticles, we have listed here. Again, we'll go over them quickly. Some of them are increased treatment efficacy, increased specific localization in the body. One of the biggest ones, obviously, is decreased toxic side effects to the patient, reduced dose, controlled biodistribution, and you can also modulate the kinetics of the release mechanisms of these different nanoparticles for drug delivery. And lastly, it improves patient compliance. The figure on the right here is a good illustration of how nanoparticles can be used to control the release rate of drugs. Here the red line is the toxic line. Below this blue line here means that the drug is ineffective. That concentration is ineffective. So we want to have, when we administer the drug to the patient, we want to have them in between these two barriers. We can design nanoparticles to have sustained release of the drug inside, as opposed to just an instantaneous degradation and release of the drugs, just spikes and dips. We can have sustained release, which is much more desirable for the patient. Again, some of the material formats. There are a variety of material formats. You can encapsulate different hydrophobic drugs using carbon nanotubes, using dendrimers, polymer particles, metal particles such as gold, particularly for diagnostics, hydrogel particles, solid lipid hybrid particles. Liposomes are extremely popular, probably one of the most popular ones, and even protein drug conjugates. Again, the size range of some of these nanoparticles are from 1 nanometer to 100 nanometers. However, a lot of the more recent articles on polymers for drug delivery recommend that the diameter of these polymer nanoparticles should be in the range of 40 or 50 nanometers. This allows them to stay in the bloodstream and can't penetrate through endothelial cells in blood capillaries and arteries and such that. However, when they come across the leaky vasculatures of tumors through their navigation through the circulatory system, they're able to leave the circulatory system through these pores, essentially these nanometer-sized pores. They're able to slip through and target the cancerous cells from within. So that's obviously a huge advantage. The surface of the outer surfaces of these nanoparticles can be functionalized, as I've said recently. You can add surface charges. You can add targeting ligands for antibodies, peptides, things that target the cancerous cells, specifically target the cancerous cells over healthy cells. That's essentially what you want. And of course, you can make these out of spheres. You can have cube architectures, rod-like architectures. There's lots of things that are possible. So here are some of the typical structures. Again, we have polymeric nanoparticles. This structure consists of drugs that are conjugated to the side chain of a linear problem with a cleavable linking bond. Some of the example compounds and polymers used in polymeric nanoparticles are PGA and HPMA. Polymeric micelles are probably one of the most popular architectures used for drug delivery applications. These are essentially amphifill block copolymers that are able to assemble and form a micellar structure with a hydrophobic core and a hydrophilic shell. PEG, pluronic, PAA, PLA, which was also biodegradable, PEG. These polymers are all used to create polymer micelles for drug delivery. Also, we have dendrimers. Dendrimers are radially emerging hyperbranched synthetic polymers with regular pattern and repeated units. You can add different types of functional groups in these dendrimers to hold on to certain hydrophobic drugs. Until a certain gradient of pH or temperature, these dendrimers can break apart and release the drugs as necessary. PAM is one of the big polymers used in this type of structure. Again, liposomes. We've mentioned liposomes. These are self-assembling closed colloidal structures composed of lipid bilayers. Lipid bilayers are used to construct cells in the body. These seem to be a great system for drug delivery. They are probably the most biocompatible architecture that we have. This is why a lot of the FDA approved nanoparticles for drug delivery are based on liposomes. These specifically can be surface functionalized with PEG, polymer PEG. Um, that's been done in several studies. There are also viral nanoparticles, and to some extent, some carbon nanotubes are being used to encapsulate some drugs for cancer delivery therapies, drug delivery therapies, excuse me. So how does this all work? What's the working principle behind this all? So the objective, of course, of targeted cancer therapy is to deliver chemotherapeutics directly to cancer tissues while minimizing undesirable toxicity to the rest of the body. So after you create a nanoparticle solution um, with these particles, you can administer them through intravenous administration, is what they refer to as. Nanoparticles are quickly distributed throughout the body by the circulatory system, and with which they are also delivered to the site of the tumor. The nanoparticles possess the unique ability to permeate through the leaky walls of the tumor vasculature, which are formed during the process of tumor growth. Through this passive targeting technique, there's also active targeting, which we'll get into, the nanoparticles can be concentrated within the tumor tissue. On the cancer cell surface membrane, the nanoparticles encounter surface receptor molecules, microscopic markers that are expressed on cancer cells, but not expressed by normal particles. So this is how the systems are able to differentiate between cancer cells and non-cancer cells, or healthy cells. So the anti-cancer drugs, once inside, can release the drug, the hydrophobic encapsulated drug, in a controlled manner by the degradation of the polymer nanoparticle shell. The highly toxic chemotherapy can thus be delivered directly to the site of the cancer. So that's the biggest advantage, obviously. Typically, the drugs will cause the cancer cells to undergo apoptosis, which is also known as programmed cell death, and the nanoparticle therapy will eventually lead to the eradication of the tumor mass completely. So that's essentially how it works. As we said, there are different types of targeting. There are passive targeting and active targeting. Nanoparticle therapeutics directly administered into the circulatory system need to be forced through the vascular walls into the target lesion and then release their payloads. So unlike small molecules, nanoparticles cannot go through tight junctions between the endothelial cells, as we mentioned, on normal vascular linings. This is owing to the fact that they are relatively large in size. However, the vessels inside a tumor region are well known for their leaky walls, allowing nanoparticles with the right sizes to pass through efficiently. These are usually around 40 to 50 nanometers. As the lymphatic system inside a tumor is largely absent or dysfunctional, the insufficient drainage facilitates accumulation of nanoparticles in the tumor tissue. This phenomenon has been widely known as the EPR effect, or enhanced passive acidity reduction effect of nanoparticles, I believe. That's what it stands for. And that's the basis for passive tumor targeting. There is also active targeting, as we mentioned. So to better utilize the biochemical properties of the cells to be targeted, ligands such as small molecules, peptides, antibodies, and antibody fragments, even nucleic acids, have been added to the surface of nanoparticles to improve their targeting efficiency. This new targeting mode that involves molecular recognition is known as active targeting, in which ligand receptor binding allows the nanoparticle to selectively and strongly bind to the surface of a specific type of cell. In our case, cancer cells, whatever they may be. And finally, drug release. There are many different types of drug release mechanisms for these smart nanoparticles. There is sustained release, diffusion controlled sustained release, and erosion controlled sustained release. We have some figures here to show you how that proceeds. Stimuli responsive release is obviously more attractive. We would like to use smart materials that are pH sensitive or enzyme sensitive, thermal responsive, photosensitive, even field responsive, to enhance the drug targeting and controlled release abilities of these particles. And again, plenty of figures shown here to demonstrate some of the different varieties of smart architectures used and how they essentially transform under different conditions to release or, in some cases, hold on to the encapsulated drugs. Future outlook, well, there are multifunctional NPs. That seems to be the future outlook. These are capable of detecting malignant cells through active targeting moieties. These can be used to visualize location of the nanoparticles in the body using real-time in vivo imaging. You can kill cancer cells with minimal side effects by sparing normal cells, of course, just like the treatments we've been talking about. And again, they can monitor the treatment effects in real time, which is extremely beneficial. The patient and essentially the doctor that's also treating the patient. So, in summary, utilization of nanoparticles as drug carriers promises a significant improvement in cancer treatments. Targeted delivery can reduce the systemic side effects that patients endure during traditional chemotherapeutic treatments by ensuring that pronounced cytotoxic levels of the drugs are only present at the tumor sites and not at healthy cells and attacking normal cells. So that's certainly undesirable. They release their payloads in response to a variety of different stimuli, either those specific to the tumor microenvironment, such as acidic pH values and elevated secretion of certain enzymes, or they can respond to external stimuli, such as electromagnetic fields. And lastly, nanoparticles can also offer multifunctionality, as we discussed briefly in the last slide, that combine both diagnostic and therapeutic features, a combination which is now being known as theranostics. So, in conclusion, that is the presentation. I hope you enjoyed it. Here for the